All right, everybody. In this week's lecture, we're going to talk about the urinary system and the physiologic mechanisms for urine formation. So, as a general overview, some of the major functions of the kidney include filtering blood, removing metabolic wastes from the blood, also regulating the water and the salt balance within the body, regulating the electrolyte balance in the body, and then regulating acid-base balance. So in terms of what's going on, here's kind of the big picture situation. Let's say this morning that we drank a protein shake. So as our digestive system begins to break down the shake, then proteins are broken down into their individual amino acids, and they're sent all over the body to repair things like our cells. While this is all great, the question is what happens to the extra amino acids that we don't use for cell repair? Well, amino acids, they're really special in that they have this compound called nitrogen in their amine group. And so over here is the amine group. And because we actually can't store amino acids as a whole, extra amino acids get processed and they get stored as being either carbs or fats. But in storing the amino acid, we don't actually need this amine group. And so it is actually converted to NH3, which is ammonia, which actually happens to be toxic to our bodies. So the liver converts this ammonia, NH3, into what we call urea. And urea is just two ammonia molecules put together. And so the liver converts this ammonia into what's called urea and packages it up and sends it to our kidneys. Now, our kidneys then will basically filter the urea, which, by the way, the liver sends this urea through the blood in a little package through the blood. And so when the blood reaches the kidneys, what happens is the urea gets filtered out of the blood and gets put into the kidneys. And so then our kidneys can excrete that urea out of the body via urine. So fun fact, once outside of the body, urea actually converts back into ammonia. And so it basically just clips the bond here, and then we end up with two NH3 molecules again, two ammonia molecules, which is also the reason why dirty pea-soaked toilets and cat litter boxes can smell like ammonia. So this is kind of the big picture of how some different things are going on in the body in terms of excreting different substances. But now we're going to turn our attention to how the kidney actually filters out the urea, for example. So what's actually going on? So urea and other substances in the blood are going to enter the kidney via the renal artery, which we know then splits into the segmental arteries and then the interlobar arteries and then the arcuate arteries. And from here, the blood is going to enter the afferent arteriole to the glomerulus. And this is kind of where the magic in the kidneys is happening, is right here at the level of the glomerulus. So the nephron is the structure that surrounds the glomerulus, and it is the structural and the functional units of the kidneys. And so basically it means that it's the tiniest structure within the kidneys that can serve functions independently. And so each kidney has millions and millions of these nephrons, and nephrons themselves are divided into two parts. So we have the renal corpuscle and the renal tubule. And so the nephron, it's kind of like the little magic wizard of the kidney. So let's remember from last week, we talked about the renal corpuscle, and we discussed that we have the glomerulus, which is that ball of looping fenestrated capillaries. And then we have the capsule that surrounds the glomerulus, also called Bowman's capsule. And remember, we said that this space has what's called the filtration membrane, which allows small substances to pass out of the blood and move into the urine. But big substances like red blood cells should stay in the kidneys, okay? Because this filtration membrane that's occurring across this prevents big substances from exiting the blood and going into the kidneys. Remember again that the renal tubule is consistent of the proximal tubule, the loop of Henle, the distal tubule, and our collecting duct. And the collecting duct, remember, is broken into the cortical collecting duct, which is up in the cortex, and the medullary collecting duct, which is down in the medulla portion of those renal pyramids. Okay, so let's talk, that was just a quick review of the anatomy. We just covered it last week, but I just wanted to refresh your memories. So let's talk about these amazing little nephron wizards. So these nephrons are responsible for 4 major processes within the kidneys themselves. And so we have the first process called filtration. And filtration occurs at the glomerulus. And what happens here is that water and solutes are going to move from the blood into Bowman's space. And once those substances are in Bowman's space, they're going to start traveling down the nephron. And as they're traveling down the nephron, a few different things occur. So, for example, in this first part of the nephron, we have a lot of reabsorption occurring. And so basically what reabsorption is it's taking substances that are still useful to us and removing them from the filtrate, so removing them from inside of the nephron and putting them back into the blood. Okay, so reabsorption is moving things back into the blood. Okay. Additionally, some other processes that are occurring along the nephron are, for example, secretion. And so certain molecules, they are perhaps too big to fit through that filtration membrane up here in the glomerulus. So what can happen is that the tubules will actually have channels that and the blood doesn't necessarily want certain substances. So the blood, these little capillaries, can actually spit out the molecules that it doesn't want and put it back and put it into the filtrate. Okay, so tubular secretion is moving things from the blood into the tubules, okay, into the filtrate. And then finally, water reabsorption. Most water reabsorption is occurring down here in the collecting duct. And so water reabsorption is just when water is going to be removed from the filtrate and put back into the blood, okay? And this is just to help regulate our water balance in the body. So say you are dehydrated, it wouldn't be wise then to urinate out extra water. So in order to preserve water, we'll actually take water that we put into the filtrate and move it back into the blood to keep our cells hydrated. Now, on the opposite side, on the flip side, if you are overhydrated, then you want to get rid of that excess water in your body. You don't necessarily need all that extra water. And so then what can happen is that water is just going to stay in the filtrate and then you're going to pee it out. Okay, so let's talk a little bit more about each of these processes in depth. So filtration, again, is the process of moving substances from the blood capillaries into our capsular space. Okay, so when fluid enters the capsular space, it is now called filtrate. So whenever it was in the vessels, whenever it was in the capillaries, it was called blood. And now the liquid that you pulled out of there and put into the capsule is called filtrate. And on average, we produce about 120 milliliters of filtrate every minute. And this is called the glomerular filtration rate. So every single minute, 120 milliliters of fluid is moved from the blood into the glomerular capsule and into the nephron. Okay, so again, this is occurring at the level of the glomerulus. But before we move on to reabsorption and secretion and excretion, we have to take a closer look here at filtration and consider the reason how we can actually filter things from our capillaries into the capsular space. So unfortunately, nephrons are not actually magical, though physics may sometimes seem like the only explanation is magic. So this is a little bit physicsy, but I'm going to try and make it as simple as possible. So the reason that we are able to move things from our blood capillaries and put them into the filtrate is due to pressure differentials between the blood capillary and the pressure that's inside of this capsular space. Okay, so there's three major pressures that we're going to talk about. And these three pressures will then create a net filtration pressure. So the first pressure we're going to talk about is the glomerular hydrostatic pressure. And basically this pressure just describes the blood pressure that's inside of this capillary. So we all know that blood pressure is the force of the blood vessel, or the blood cells against the walls of the blood vessels. And so inside of these capillaries, there is fluid, and that fluid is pushing up against the walls of these capillaries, and that's what's creating this glomerular hydrostatic pressure. Now, on average, the glomerular hydrostatic pressure is approximately 55 millimeters of mercury. Okay, so this pressure is responsible for pushing fluid and for pushing solutes out of the glomerular capillaries and into the capsular space. All right, so that this particular pressure is going to want to move stuff from the capillaries into the capsular space. Okay. The capsular hydrostatic pressure is also a pressure that is going to be acting on this process of filtration. But basically, the capsular pressure is going to be the force that's created by the fluid that's within this capsular space. Okay, so if you're pushing fluid from the capillaries into this other space we call the capsular space, then you are going to effectively be creating some pressure, right? So if some stuff is in here, it's also going to be pushing against those capillaries. Okay, so hopefully that makes sense. But this poses a sort of opposite effect as the glomerular hydrostatic pressure. So the capsular hydrostatic pressure has a value of about 15 millimeters of mercury, and it actually opposes the filtration. So it's working against the glomerular hydrostatic pressure. So this particular pressure is going to be moving from the capsular space and pushing stuff back into the capillaries. Okay, so it's going to want to push stuff back into the capillaries. All right. This third pressure that we're going to talk about is also a pressure that's inside of the capillary. So inside of the capillary, we already said, so this is a capillary, remember, it's just a little tiny blood vessel. We said we have this pressure, the blood pressure essentially, that's going to be pushing stuff out. Okay, that's absolutely true. However, we also have a pressure in this capillary that's going to be created by proteins that are in the blood and other solutes that are in the blood that basically works like a vacuum pressure, sucking things in. Okay, so if, for example, I don't know, salt is trying to leave, you're going to have substances that are in here acting kind of like a tug of war situation, trying to pull that salt back in. And that pulling pressure, the sucking pressure, is going to be referred to as our colloid osmotic pressure. And so if this is a pressure that's going to be pulling stuff, trying to keep stuff in the capillaries, this one's going to oppose filtration, right? Because the filtration is referring to moving stuff from the capillary out to the space. If we're trying to keep stuff in the capillary, that's going to oppose filtration. And just to give a numerical value here, the colloid osmotic pressure then has a value of about 30 millimeters of mercury. Okay, so here's the math fun part. All right. So let's talk glomerular hydrostatic pressure. If we have, let me just draw this out for you guys. If we have a pressure, and we're going to say that this side is capillary, and then we're going to say over here is nephron, then here's what's going to happen. Okay, our glomerular hydrostatic pressure is going to be pushing stuff from the capillary into the nephron, right? And we're going to measure this value at 55. Okay, so this is pressure number 1. Pressure number 2, we said, was the capsular hydrostatic pressure. And this one is working against filtration. So it's pushing water back into the capillaries, and it's about 15. So pressure number 2, now we're moving stuff back into the capillaries. So it's going to go that way, right? It's opposing this first pressure. Okay. Pressure number 3, we said, is this colloid osmotic pressure. And this is sort of like that vacuum pressure that's trying to keep things in the capillaries, right? So this one is a pressure of 30 millimeters of mercury, keeping stuff in the capillaries. So let's add that. All right, so we're going to say that's 30. And that was pressure number 3. Okay, so what are we left with? We are left with one pressure moving that way at 55, 2 pressures moving this way at a total, when you add those two together, of 45. Therefore, our net filtration pressure is going to be this segment right here, which we can see is going to be moving from the capillary to the nephron at about 10, right? Because 55 minus 45 is about 10 millimeters of mercury. So our net filtration pressure here is moving from capillary to nephron at about 10 millimeters of mercury. This is why we are filtering. Okay, this is the only reason why we are filtering, is because there is a net pressure pushing stuff out of the capillaries towards the nephron. If either of these pressures here increase, or if this first pressure decreases, it got smaller, filtration might not occur, okay? So if you have any questions about this, please feel free to contact me via email or Remind or Signalvine. So now that we've talked about filtration, let's talk about reabsorption. Reabsorption then is the process. So now we put stuff into the nephron and we're starting to move that stuff down along this nephron structure. All right, so reabsorption then is the process of taking stuff that we have in the nephron and the filtrate in here and putting it back into the blood. So why would we do this? Well, remember, some things, really small molecules, for instance, can slip through the filtration membrane because they're so small. They're going to go in there no matter what. But we don't necessarily want to get rid of them. It just so happened that they were small enough to fit through that filtration membrane. So this is how we get some of that stuff back that we didn't mean to lose. So things like salts, like bicarbonate, like water, things like glucose and amino acids. Okay, so this process of reabsorption is moving stuff from the nephron, from inside the nephron, the filtrate, back into the blood. And this is actually occurring all along the capillary. Now, I want to point out something really important here, that is water. Okay, so we notice that there is water being reabsorbed all along the nephron here, right? And this is very, very valuable to us because if we did not reabsorb water in the tubules or in the collecting ducts, we would lose our entire plasma volume, right? And remember, plasma is that top layer of the blood when we spun it around in a test tube. Remember, we had red blood cells on the bottom, and then we had a little buffy coat layer, and then we had plasma. Remember this plasma layer. So if we did not reabsorb water, the entire plasma volume, all of this would be gone in fewer than 30 minutes. So it's very, very important that we are able to reabsorb water along the nephron. All right, so the opposite process of reabsorption is secretion. And secretion is when we are going to move substances from the blood in the peritubular capillaries back into the filtrate. So along the nephron, we know we have the glomerulus here, but we're not talking about secretion here, okay, because this is filtration. We're talking about secretion in the sense that all along this area, we have capillaries that are wrapping up the nephrons. And these are called the peritubular capillaries, okay? So they're sort of wrapping up then the nephrons everywhere, okay? I just drew them here so they would be out of the way. But the process of secretion is when we have some stuff in these capillaries that we need to get rid of, we can just simply transfer it from these capillaries over into the nephron. So some of the major things that we secrete are things like potassium ions and hydrogen ions. These are important for maintaining our acid-base balance. And then also uric acid is something that we will secrete. And in terms of secretion, where does it occur? Remember, we said reabsorption is kind of occurring everywhere, but secretion mostly is only occurring at the proximal and the distal convoluted tubules. Okay, so really only occurring up here in the cortex of the kidney. So where then what is the process then, I should say, for that urine is going to take? So we're going to make the urine up here, and then we're going to send it to the collecting duct. Oops, I'm sorry, I keep flipping that, I don't mean to. Okay, so we're going to make urine up here in the nephron, and then it reaches the collecting duct. From the collecting duct, so then we're going to come down here. The urine is going to be drained into our minor calyces, and then our major calyx, and then our renal pelvis, and then our ureter. And then our ureters are going to take that urine and dump it into the bladder, which then the urine will go to the urethra, and then finally the toilet. So now we're going to talk about this process. So we've already talked about kind of everything that's happening in the kidney. Now let's talk a little bit more about what's happening beyond the kidney until we reach the toilet part, okay? So the term excretion then is the process of moving substances from the nephron tubules to the outside of the body in a process we call micturition. And micturition is just the fancy word for urination. And so urine in general is this aqueous solution of both organic and inorganic substances. And urine alone is about 95% water, okay? And then about 5% solutes. And of those solutes, we have both organic solutes and inorganic solutes. So organic are things that would contain carbon, nitrogen, that sort of group of chemicals. And then inorganic is basically every other element on the periodic table. So some organic components that are present in urine, first and foremost, the most numerous organic component is urea. You can see that urine is about 2% urea. And urea, that word should sound familiar to you because we talked about it when I went over the big picture slide. So urine is about 2% urea. And then it also has some creatinine and some uric acid. And creatinine is a byproduct of muscle metabolism. And then uric acid actually is a product of nucleic acid catabolism. And so your nucleic acids are your DNA and your RNA. So uric acid comes from the catabolism of nucleic acids. And this uric acid aspect is important because this is the one that tends to form crystals. And this is the one that if those crystals are formed, they can build up and become what we know as kidney stones. So some of the then inorganic components are things like sodium chloride, which is salt, and this is the most abundant mineral salt. And the amount that is in the urine, it just depends on how much salt you have in your diet. Potassium ions are also present, and sulfates and phosphates are also going to be the inorganic components that are present in the urine. Now, I just want to point out one more thing kind of going along with this, and that is that if you remember back to when we were talking about glomerular filtration rate, and that's about 120 milliliters per minute, and it's due to the net filtration pressure. Now, if that filtration pressure is too high, or if the GFR is too high, so say maybe it's 200 milliliters per minute, then what's going to happen is our nephrons aren't going to have time to reabsorb all of the necessary substances in our filtrate. And this is the case in which much of our water and other important solutes would be lost in the urine. And so we would have varying concentrations of our important solutes here in the urine. So too high of a pressure is a bad thing, right? So this is also one of the reasons why people with high blood pressure can develop kidney disease, because the kidneys are very sensitive to blood pressure. And so when the pressure is too high, that increases the filtration rate, and then you start losing things in your urine. In addition, high pressure situations also are not good for your vessels. And because the kidney is so sensitive to high pressure situations, they begin being unable to regulate the blood pressure within the kidneys, and too high of a pressure basically just destroys our nephrons. Okay, so now that we've talked a little bit about urine, I just made a couple of tables for you guys, and this is going to be useful on your exam, of first and foremost the physical characteristics of normal urine. So normal urine volume should be between 0.7 and 2.5 liters a day. In terms of color, it should be a pale yellow color, but if you are dehydrated, then you will have more solutes, less water, and so that will make the urine a little bit darker. In terms of turbidity, basically that means the sort of clearness, your ability to sort of see through it. Whenever urine is fresh, it should be relatively clear. It should be sort of so you can look through it. But after urine has been standing for a while, it becomes turbid, which means it becomes kind of cloudy. In terms of odor, it should have an aromatic odor, sort of like that's when it's fresh, but then after it's been sitting for a while, we know that urea, remember, breaks back down into ammonia. And so the ammonia-like odor will be present after urine has been standing due to that bacterial breakdown of urea. In terms of pH, so pH measures how acidic or how basic the urine is. Normal pH of urine should be between 4.8 and 7.5, but the average is about 6.0. And so if you guys know anything about pH, then we should know, hopefully, that our pH scale kind of looks like this, and 7 means neutral. Anything above 7 is going to be basic. Anything below 7 is acidic. So we can see then normal, our average urine then is slightly acidic with a pH of about 6. However, if you have a high protein diet, then we know that proteins break down into amino acids. It literally says the word acid in it, so it's going to make your urine even more acidic. However, if you eat a lot of vegetables tend to be more basic and or alkaline. And so if you eat, say for example, you're a vegetarian, then vegetarians would tend to have a more alkaline, maybe a more basic pH. So in terms of specific gravity essentially just measures concentration of solute compared to concentration of water. Okay, so let's think about it like this. I'm going to just draw a little picture. If we have a beaker, be beaker 1, and it has some water in there, the specific gravity of only water, just water, is 1. If we have another beaker, we have some water in there, but say we also have some solutes in there. Say we have like some salt, for example. We put some salt in that water. This means that our specific gravity is going to be greater than 1. And this should make sense for urine, because remember I said that urine was about 95% water and about 5% solute. And so that would mean that the specific gravity of urine should be greater than 1. Not a lot greater, right? Because there's not very many solutes in there when you compare that with water, but it should be higher than what water's specific gravity is. If, for example, let's kind of compare this clinically then, if you are dehydrated, that would mean that you have more solute in your urine because you're trying to preserve water, so you're going to pee out less water. This would make it so that the specific gravity would be even higher, right? So maybe now you have 10% solute and only 90% water. But if you're overhydrated, then that specific gravity would be closer to the value that water has. So these are just some pathophysiological conditions. So I'm going to have you guys just read over these yourself. The only one I do want to point out is going to be this one right here. Diabetes in terms of odor. People with diabetes tend to have fruity smelling urine. And this is one of the sort of easiest ways to kind of jab at the idea that diabetes might be present, is because of this fruity odor. So in diabetic patients, what tends to happen is they tend to have really high blood sugar. Well, what's a great way to get rid of extra sugar in your blood? You can just put it into the urine. So the presence of sugar, all this extra sugar in the urine, gives urine this fruity smell. And then here's just, again, one more slide for you guys to look at on your own time. So that concludes the lecture for the physiology of the kidneys, and let me know if you guys have any questions.